previous class we explained about virus up to symmetry. Now we discuss virus remaining and plant physiology chapter water relations. Virus remaining means previously we discussed up to capsomere arrangement symmetry. Now we discussed this structure of bacteriophage, structure of tobacco mosaic virus, replication in viruses, then how viral diseases in plants and how viruses are transmitted and how virus is controlled. So, coming to the first one, we will go to the bits. First bit, the nucleic acid in blue tongue virus is. On that day I told normally in viruses there will be double stranded DNA or single stranded RNA. Double stranded DNA is normally present, but some shows single stranded DNA. They are 5,7 into 174 bacteriophages, 5x into 174 bacteriophages and M13 bacteriophages. There are some with double stranded RNA. They are reovirus, wound tumor virus, reovirus, wound tumor virus, blue tongue virus, maize rough dwarf virus, maize rough dwarf virus, rice dwarf virus and others. So, in blue tongue virus the genetic material is double stranded DNA, double stranded RNA. So, nucleic acid in blue tongue virus is double stranded RNA, second option. Next, protomers are absent in. Protomers means capsomeres. The other name of protomers is capsomeres. So, protomers are absent in not wound tumor virus, because wound tumor virus is a complete virion with nucleic acid and capsomeres. Rice dwarf virus. Rice dwarf virus is also a virus which is a complete virion with nucleic acid and capsid protein coat. M13 virus. M13 virus is also a complete virus with protein coat and nucleic acid. In this, potato spindle tuber virus. Potato spindle tuber virus is a viroid. What is viroid? It shows only nucleic acid. A protein coat is completely absent. If protein coat is completely absent, capsomeres are absent, that is protomers are absent. So, protomers are absent in potato spindle tuber virus, second option. In TMV, the structure with 4 nm size is. In TMV, the structure with 4 nm size is, 4 nanometer size is. The central core, it includes single stranded RNA. Around this single stranded RNA, capsomeres are arranged in the form of a spiral staircase. So, they are arranged in the form of a spiral staircase. These are capsomeres, this is central core, and within the central core you will find single stranded RNA. The speciality of central core is it is open on both sides. It is a rod shaped virus. So, here the question is in TMV the structure showing 4 nanometer size is central hole, length of particle, diameter of particle, diameter of capsomere. So, central hole length is around 300 nanometers, diameter is around 18 to 19 nanometers, diameter of capsomere is not there. So, central hole, the central core or central hole is around 4 nanometers size, first option. The total number of nucleotides in the RNA of tobacco mosaic virus. The total number of nucleotides in RNA of tobacco mosaic virus. The total number of capsomeres are 2130. The molecular weight is 39 into 10 power 6 Daltons. Each capsomere contain 158 amino acids. Then the nucleotides in RNA will be 6500. So, capsomeres are 130. Molecular weight is 39 into 10 power 6 Daltons. Each capsomere contains 158 amino acids, and above all, the number of nucleotides in the RNA of TMV, not 6000, not 2130, that is the capsomere number. 39 into 10 power 6 Daltons is molecular weight, 3000 is not there. So, answer is 6500. So, the total number of nucleotides in the RNA of tobacco mosaic virus is 6500 nucleotides. This is first option. The minimum number of nitrogen bases present in the gene of TMV that code its protomer. That code its protomer is protomer contain 158 amino acids. 158 amino acids means 158 into 3 you have to write. If you write 158 into 3, some number will come. To that number, if you add 3, you will get the minimum number of nitrogen bases present in the gene of tobacco mosaic virus that code a protomer, that code a capsomere, that code a protein in capsomere. The answer is 477. So, the minimum number of nitrogen bases present 158 amino acids into 3 plus 3. Why plus 3 means we have to write anticodons also. The total number of nitrogen bases present in the gene of tobacco mosaic virus that codes its protomer is 477. The third option is for this 477. Next. Each capsomere of tobacco mosaic virus covers about the following number of ribonucleotides. Each capsomere of tobacco mosaic virus, each capsomere of tobacco mosaic virus covers about the following number of ribonucleotides. Ribonucleotide number is 6500. The capsomere's number is 2130. So, it is around 3. So, answer is 3. Each capsomere of tobacco mosaic virus covers about the following number of ribonucleotide means 3, around 3 ribonucleotides. First option. Bacteriophages were first studied in detail. Bacteriophages were discovered by Twart. They are studied in detail by D. Harreley. He was the scientist who proposed that every bacteriophage has a hexagonal head and a narrow tail. Hexagonal head and a narrow tail. In between head and the tail there is a structure called collar whose function is not known. Tail is surrounded by a capsid protein coat. Head is surrounded by a capsid protein coat. Tail is surrounded by a capsid protein. Head includes double-stranded DNA. At the base of the tail, there are 6 spikes. 6 spikes produces 6 tail fibrils. The core of tail is empty, but the core of head is filled with double-stranded DNA. So, this type of structure, tobacco mosaic virus, was first given by Felix D. Harreley. So, bacteriophages means the virus which attack bacteria, and bacteriophages are studied in detail by means Felix D. Harreley, not Twart, not Delbruck. Delbruck is known for lytic cycle in viruses, Boden and Pyre chemical nature of viruses. So, Felix D. Harreley will be the answer for this. In a cross section, the head of bacteriophages. In a cross section, head of bacteriophage is not pentagonal, not triangular, not quadrangular. It is hexagonal. It has 6 faces surrounded by capsid protein coat, central core with double-stranded DNA, and capsid protein coat made up of capsomeres. Capsomeres are arranged in a cubical symmetry. That means they are arranged in the form of a cube. So, in a cross section, the head of bacteriophage is hexagonal in shape. Fourth option. The head protein of bacteriophage is formed the following type of membrane. The head protein of bacteriophage is made up of which of the following protein? It is impermeable, it is permeable, it is semi-permeable, it is selectively permeable. Answer is semi-permeable. The head protein of bacteriophage is formed the following type of membrane means it is semi-permeable membrane, that is a plasma membrane. Head protein is bacteriophage is formed by a semi-permeable membrane, that is plasma membrane. Third option. Prophase is based on the life cycle. there are two types of viruses. One is virulent phase, the other is prophase or temperate phase. Based on life cycle, viruses are divided into two types, prophase and temperate phase. Virulent phase means virus enters the bacterial cell, does not incorporate with bacterial DNA, does not incorporate with bacterial DNA, cause lytic cycle. That means immediate breakdown of bacteria is done by virulent phases. Prophase is one which enters the bacterial cell, fuses with bacterial DNA, and along with bacterial DNA replicates for many generations. Any bacterial cell whose DNA is incorporated with viral DNA is called lysogen. Any bacterial cell whose DNA is incorporated with viral DNA is called lysogen. That's why this cycle is called lysogenic cycle. So that is the difference between virulent phase, prophase or temperate phase. Any virus which is having the capacity of forming prophase is also called temperate phase. So virulent phase causes lytic cycle. Temperate phase or prophase causes lysogenic cycle. Immediate breakdown of bacterial cell to release viruses is lytic cycle. They remain many years along with bacterial DNA, and after some years they get separated from bacterial DNA and shows its independent life, and then breaks the bacterial DNA is called lysogenic cycle. It is done by prophase. So here prophase is DNA of a virus that cannot integrate with the host DNA is wrong. First form virus is wrong. The first bacteria phase formed in bacterial cell during reproduction is wrong. DNA of virus that can integrate into host DNA is called prophase, and the capacity prophase which can convert it into this special structure is called temperate phase. So answer is DNA of virus that can integrate into the host DNA. Second option. Viral replication is understood by the experiments of. Viral replication is understood by the experiments not by Twart, not by Bazerink, not by Franklin, because Twart and D. Harrelly bacteriophages. Bazerink is known for contagium, vivum fluidum or virus particle. Franklin RNA is the genetic material in plant viruses. Delbruck, viral replication is understood by the experiments of Delbruck. This is answer, first option. The action of enzyme lysozyme is seen in during the following stage of viral reproduction. During viral reproduction there are four important stages. One is adsorption stage. The other is penetration stage. The third is latent stage or latent period. Last is lytic stage or lysis. During adsorption stage, a bacterial DNA goes and attaches to the surface of the bacteriophage goes and attaches to the surface of the bacterial DNA and attaches to the surface by means of tail fibrils. Next, the tail fibril secretes a special enzyme called lysozyme, which drills the bacterial cell wall to make a small pore. Now contraction of membrane protein takes place. The viral DNA is incorporated into the bacterial DNA. So the activity of lysozyme comes when the bacteriophage goes and attaches to the cell wall of bacterial cell by what is called tail fibrils. Once the tail fibrils attaches, lysozyme is secreted, small pore is made, and when viral DNA contracts, the DNA is incorporated into the bacterial cell. So the action of enzyme lysozyme is seen during the following stage of viral reproduction is not adsorption. Adsorption is A tail fibril go and attaches to the surface of the bacterial cell. Latent means, latent is a period which is observed in two stages, eclipse stage and maturation stage. Eclipse stage means nothing appears in the bacterial cell after the entry of viral DNA. Maturation stage means new viral particles are formed. So, this is also asked as name the lytic cycle or name the stage in lytic cycle of viruses where bacteria appears like a factory of viruses. Listen carefully. Name the lytic cycle of viruses where bacteria appears as a factory of viruses is maturation stage. So, action of enzyme lysozyme is seen during the following stage of viral reproduction means penetration stage. For penetration, small pore has to be made and that pore is made by lysozyme and that stage is called penetration stage. The first step in during replication is, first step is always attachment of bacteriophage to the surface of bacterial cell wall with the help of tail fibril. So, recognition of host cell is wrong. Lysozyme action occurs after that. Degradation of host DNA occurs only after the entry of viral DNA into bacterial cell. So, collisions between bacterium and viruses is the first step in viral replication. A bacteriophage goes and attaches to the bacterial DNA at a particular place. This is called collision, and collision is always irreversible. That is, only viruses goes near the bacterial cell. Bacterial cell does not come nearer to viral. So, collision between bacterium and viruses is the first step during viral replication. In one of the following stages of viral replication, viral reproduction is not noticed. In one of the stages of viral replication, viral reproduction is not noticed. It means first adsorption, a bacteriophage comes and attaches to the bacterial cell with the help of tail fibril. Later, tail fibril secretes a special enzyme lysozyme which is making a small pore in the bacterial cell wall. Penetration takes place, bacterial DNA or viral DNA enters the bacterial cell. Whenever viral DNA enters the bacterial cell, the bacterial DNA disintegrates. Once the bacterial DNA disintegrates, the total activity of viral or bacterial cell comes under the hands of viral DNA. Now, viral DNA starts dividing and ultimately produces new viruses. But before that, in the latent stage, a stage called eclipse stage, where you do not find anything in bacterial cell also. So, in one of the following stages of viral replication, viral reproduction is not noticed is eclipse stage. The eclipse stage is the out of two stages in latent stage. Latent stage is known for, one is eclipse stage, the other is maturation stage. Here lytic stage is wrong, penetration stage is wrong, adsorption stage is wrong. Answer is latent stage, second option. Infectious particle has only nucleic acid is. Infectious particle having only nucleic acid means it has only genetic material without capsid protein code. So, citrus exocartosis virus, rice dwarf virus, tongue row virus. Rice dwarf virus, tongue row virus are complete viral particles. Citrus exocartosis virus is a viroid. Viroid means only genetic material is present, but capsid protein coat is absent. Capsid of virus helps in. Capsid of virus means capsomeres made up of capsomeres. Capsid protein coat of viruses is helping in protection to nucleic acid, transferring the nucleic acid from host cell to other and taking over the functions of host cell. Taking over the functions of host cell is wrong. It protects the nucleic acid. Definitely it is a covering layer of nucleic acid, so it protects the nucleic acid. Transferring nucleic acid from host cell to other is wrong. It is correct. It always transfers. When it contracts, the nucleic acid present in virus enters the bacterial cell. So, transferring the nucleic acid from host cell to other. So, first and 2, 1 and 2 are correct for capsid of virus helps in protecting nucleic acid, transferring nucleic acid from host cell to other. Plant virus with DNA as genetic material. So, normally plant viruses has RNA. Animal viruses have, plant viruses have RNA, animal viruses have DNA. But there are some exceptions. Cauliflower mosaic virus, Dahlia mosaic virus. cauliflower mosaic virus. Dahlia mosaic virus contains DNA. Influenza virus and polio virus has RNA. So, animal viruses with RNA are influenza virus and polio virus. Plant viruses with DNA are cauliflower mosaic virus and Dahlia mosaic virus. These are the two plant viruses with DNA. So, plant viruses with DNA as genetic material are, one is Dahlia virus, the other is cauliflower, Dahlia mosaic virus and cauliflower mosaic virus. So, 1 and 3 will be the answer. Arrange the following events in viral reproduction in a correct sequence. Breakdown of bacterial cell, synthesis of viral DNA, ghost formation, attachment of virus to bacterial cell. Anytime the first step is, this is the bacterial cell, a virus comes and attaches to bacterial cell with the help of its tail fibers. Tail fibers produces an enzyme called lysozyme, which makes a small pore in the bacterial cell, and DNA is incorporated into the bacterial cell, and that is called penetration stage. Now, the empty hexagonal head formed after that is called ghost. The empty hexagonal head of virus formed after that is called host. So, breakdown of bacterial cell, synthesis of viral DNA, ghost formation, attachment of virus to bacterial cell. Here the first step is attachment of virus to bacterial cell. 4. Next, ghost formation. Once attachment occurs, penetration takes place, the viral DNA enters bacterial cell, resulting in the formation of ghost. Then synthesis of viral DNA, because once the viral DNA enters bacterial cell, bacterial DNA disintegrates and every total metabolic activities are under the control of viral DNA. So, synthesis of new viral DNA takes place. Once the number of viruses reaches to certain extent, the average number of viruses in a bacterial cell just before the bacteria is going to break is called burst size. So, the stage in bacterial or viral reproduction where the number of viruses reaches saturation and the bacterial DNA starts breaking is called as burst size. So, here the answer is 4, 3, 2, 1. Arrange the pathogens in a sequence that have only nucleic acid and protein coat, only protein covering, nucleic acid, capsid and peplose. That have only nucleic acid is citrus exocartosis virus. That have nucleic acid and protein coat is tobacco mosaic virus. That have only protein covering is citrus, I don't say citrus, it is a scrapie disease in sheep, virion, and nucleic acid, capsid and peplose is influenza. So, 1, 3, 2, 4 is the answer for this. Arrange the following events of virus growth cycle in a sequence. Action of lysosome on bacterial cell wall, role of tail fibers in viral growth cycle, synthesis of viral DNA, synthesis of capsid protein coat. First, role of tail fibers in viral growth cycle. Tail fibrils goes attaches to bacterial cell wall superficially, then they secrete lysozyme. Then action of lysozyme on bacterial cell wall. Once the tail fibril attaches, then action of lysozymes is observed on the bacterial cell wall. Then synthesis of capsid. First capsid protein coats are produced, then synthesis of viral DNA. So, answer 2, 1, 4, 3. First two, role of tail fibers in the viral growth cycle. Second, action of lysozyme. Once the tail fibrils are attached, then lysozyme action starts. So, action of lysosomes and lysozyme on bacterial cell wall. Then synthesis of capsid protein coat. First capsid protein coat is formed, then synthesis of viral DNA occurs, new viruses are formed. So, 2, 1, 4, 3 will be the answer. Match the following: adsorption, penetration, latent period, lysis. This is also regarding lytic cycle. Synthesis of coat proteins, intracellular lysozyme action, collision between virus and the bacterium, dissolution of protein coat, lysozyme action by extracellular viruses. So, here the answer is A3, adsorption. collision between virus and bacterium. B5, penetration, lysosome action by extracellular viral particle. Latent period, synthesis of new coat proteins. Synthesis of new coat proteins is latent period. Lysis, dissolution of protein coat. When dissolution of protein coat takes place, or intracellular lysosome action. Intracellular lysosome action takes place, all the viruses produce lysosome, bacterial cell breaks and all the viruses are released out, that is called lysis. So, answer is adsorption 3, collisions between viruses and bacterium. Penetration 5, lysosome action by extracellular virus, external virus. Latent period, synthesis of coat proteins, and lysis, it is intracellular lysosome action. All the viruses produces lysosome which breaks the cell wall and all the viruses are liberated out, that is called lysis. Match the following: Oryza, Nicotiana, Abelmascus, Lycopersicum. So, these are the diseases caused by viruses on these plants: vein clearing, ring spots, vein banding, mosaic, chlorosis. For this, the second option, Oryza, chlorosis. Oryza is chlorosis. Nicotiana B, mosaic. We all know tobacco mosaic disease. Tobacco is Nicotiana. So, Oryza is chlorosis. Nicotiana is mosaic. Abelmascus, bendy vein clearing. It is popularly called bendy vein clearing. So, Abelmascus is vein clearing. And Lycopersicum is ring spots. Ring spots in Lycopersicum. Tomato ring spot virus we call. So, tomato is Lycopersicum. So, Oryza, chlorosis. Nicotiana, tobacco mosaic disease. Abelmascus, vein clearing, bendy vein clearing, citrus vein banding. There are several diseases by viruses. So, vein clearing. And Lycopersicum, ring spots. Tomato spotted wilt virus. The name of the virus is tomato spotted wilt virus, which causes the formation of circular spots. Match the following: fungi, seeds, insects, pollen grains. This is the transmission: cherry ring spot, papaya mosaic, potato virus, lactuca mosaic, and tobacco necrosis virus. A5, fungal, TNV, tobacco necrosis virus is transmitted by motile juice spores of a special fungi called Olphidium brassicae. Tobacco necrosis virus is transmitted by motile juice spores of Olphidium brassicae. So, fungi is tobacco necrosis virus. Seed is lactuca mosaic virus and lettuce mosaic. Lettuce mosaic and lactuca mosaic both are transported or transmitted or transferred by seeds. Insects, papaya mosaic virus. Insects always papaya mosaic virus, rice tungro virus, and spotted wilt virus also by insects. Pollen grain, the only virus which is transmitted by viruses during pollination is cherry ring spot virus. So, answer is 3. A5, fungi, tobacco necrosis virus, motile juice spores of Olphidium brassicae. Seeds, lactuca mosaic and lettuce mosaic both are transmitted by seeds. Insects transmit hundreds and hundreds of viruses, but the most popular ones are papaya mosaic virus. Pollen grain always transmits cherry ring spot virus. Again the same matching type, but here a little bit different. Study the following 2 columns and match the items correctly. List 1, lettuce mosaic, papaya mosaic, cherry ring spot and soil borne viral diseases. Xiphinema, pollen grain, seeds, mechanical methods and insects. For this, answer is 4. A3, lettuce mosaic virus is by seeds. B5, papaya mosaic virus is by insects. C2, cherry ring spot virus is always by pollen grains, and soil borne viral pathogens diseases are transmitted by special nematodes called Xiphinema, Longidorus, etc. So, answer is 4. Prophase. Prophase is DNA virus that can integrate into host DNA, that is prophase, which is also called temperate phase. Stage of lytic cycle, cell event 1 and event 2. Cycle event 1 and cycle event 2. Here we have to identify the correct answer. Penetration says cell wall dissolution, contraction of viral tail sheath is correct. Adsorption stage, lysozyme action, recognition of bacterium. Recognition of bacterium is correct, but absorption of or lysozyme action is wrong. Adsorption does not show lysozyme action. Latent period, coat protein synthesis, assembly of DNA and capsid viruses is correct. Latent stage is coat protein synthesis, assembly of DNA and capsid virus is correct. Lysis, lysozyme action, lysis of virus, wrong. Lysis, lysozyme action is correct. Not lysis of virus, it is the lysis of bacterial cell to liberate or send out viruses. Answer 2. 1 and 3 are correct, 2 and 4 are wrong. Malformation, excessive growth of stem, cocoa. Floral breaking, loss of color by petals, tulip. Ring spots, loss of color on the leaf in the form of circles, cherry. Chlorosis, discoloration of the stem, rice. So, correct combinations are malformation, excessive growth of stem, this is called swollen shoot in cocoa, first is correct. Floral breaking, loss of color or addition of colors or something like that in tulip is correct. Ring spots, loss of color on the leaf in the form of circles, that is why ring spots, it is correct, cherry ring spot virus. Chlorosis, discoloration of the stem, rice is wrong. Chlorosis is discoloration of the leaf, not stem. So, 1, 2, 3 are correct, fourth one is wrong. Tobacco mosaic virus has helical and adenovirus has cubical symmetry. Arrangement of capsomeres is entirely different in tobacco mosaic virus and adenovirus. In tobacco mosaic virus, protein capsomeres are arranged in helical manner, spiral staircase manner. In the case of adenovirus, they are arranged in the form of cubical manner. Each capsomere is a equilateral triangle face. Such 20 faces are arranged, which is also called icosahedron symmetry. So, first option for this. Influenza virus has capsid and peplose. All RNA viruses contain envelope. Influenza virus is an animal virus which has capsid protein coat. Around the capsid protein coat there is a peplose. Reason, all RNA viruses have envelope, it is wrong. We cannot say that all RNA, influenza virus is absolutely a virus with DNA, not RNA. So, all RNA viruses have envelope is wrong. So, A is correct, R is false. A is true, R is false. Polio virus has RNA. Plant viruses usually have RNA. Polio virus have RNA is correct, because influenza virus and polio virus are animal viruses with RNA, but plant viruses usually have RNA. So, any plant virus will definitely contain RNA. Because plant viruses have RNA, polio virus does not have RNA. So, polio virus having RNA is correct, plant viruses usually having RNA is correct, and because they are not related to each other, the answer is second option. A and R are true, R is not the correct explanation of A. So, these are some of the questions we discuss about the control also. So, control of viruses, 1, exclusion of pathogen, exclusion of inoculum. That means the pathogen, once a plant is seen with a disease, it is completely eradicated, or special quarantines are taken into consideration to prevent the entry of pathogen. Then eradication of pathogen. Eradication of inoculum is complete removal of virus. This removal of virus by burning the infected plants is popularly called roguing. Then chemotherapy. Chemotherapy means various chemicals are used like cytovirin 2, thiouracil, malachite green, 2,4-D. 2,4-D is 2,4-dichlorophenoxyastic acid. It is both weedicide and viricide. So, 2,4-D is both weedicide and viricide. Now, finally, growing disease resistant varieties. whenever disease resistant varieties are grown, then new plants will not get the disease. So, the best method to control any viral disease is growing disease resistant varieties. These are some of the control measures of viruses. One is exclusion of pathogen by strict quarantines. The entry of pathogen is minimized. The other is eradication of pathogen. Whenever pathogen is supposed to be there in the plant, the total plant must be completely burnt off. Then this is called roguing. Then chemotherapy. So many chemicals are used to control various viral diseases. Then exclusion of, or what you call 2,4-D and other things, and the best is growing disease resistant varieties. That's about microbiology, bacteria and virus. Now we will enter plant physiology. We discuss water relations. Water is popularly called elixir of life or liquid of life. Water is popularly called elixir of life or liquid of life. 90% of protoplasm is water. Seed germination occurs in the presence of water. Translocation of food metal occurs in the presence of water. Photosynthesis requires water. Respiration requires water. Loss of water in the form of water vapor is transpiration. Transpiration is responsible for the absorption of water by roots. So, water is very important in the life history of plants. Okay, we discuss some important bits. One, free energy of a substance with which it moves or involved in chemical reaction is called. What is free energy? When potential energy of a component is converted into kinetic energy, some amount of energy is liberated, which is called free energy. Whenever free energy is liberated, that free energy is utilized to complete a reaction, make some water to movement or anything. This free energy present in 1 mole of a substance in any form. 1 mole of a substance in any form is called chemical potential. Chemical potential. The chemical potential of water is called water potential, which was denoted by a symbol psi, and the term water potential was given by Slater and Taylor, and it is measured in megapascals. So, whenever the potential energy of a substance is converted into kinetic energy, definitely an amount of energy is liberated, which is popularly called free energy. This free energy is useful to perform any step or reaction in anywhere. The free energy present in 1 mole of a substance in any form is called as chemical potential. The chemical potential of water is called as water potential. So, free energy of a substance with which it moves or involved in various chemical reactions is not called osmotic potential, pressure potential or metric potential. It is simply called as chemical potential. First option. The amount by which water potential of a solvent decreases due to the addition of a solute is called. When a pure water is taken, pure water's water potential is always 0. When something is added to pure water, that is solute is added to pure water. Whenever solute is added to pure water, its concentration increases. Its concentration increases. Whenever concentration increases, water potential decreases. So, water potential and concentration are inversely proportional. Water potential decreases, that means free energy decreases. The amount of decrease in the water potential of pure water, the sum of decrease in the water potential of pure water due to the addition of a solute, is popularly called solute potential or osmotic potential, denoted by a symbol psi pi, and its value is always negative, and it is also measured in megapascals. So, the question is, what is the question here? The amount by which water potential of solvent is pure water, decreases due to the addition of solute is called not metric potential or pressure potential, but it is osmotic potential, not turgor potential. So, answer is osmotic potential. This is 0 in plasmolized cell. Whenever a cell is kept in hypertonic solution, whenever a cell is kept in hypertonic solution, whatever water present in the cell moves out, because water moves from a region of higher water potential to a region of lower water potential, that is from a region of lower concentration to a region of higher concentration. So, whenever water moves out of the cells, 90% of protoplasm is water, so shrinkage of protoplasm occurs, which is called plasmolysis. In a plasmolysis, water potential is equal to pi plus 0. Water potential, pi is osmotic potential, 0 is pressure potential. That is why the value of water potential is exactly equal to osmotic potential. So, according to this question, in a plasmolized cell, this is 0 in a plasmolized cell is not water potential, not solute potential, not metric potential, but pressure potential. Because pressure potential is 0, the water potential value is exactly equal to solute potential. So, fourth option, pressure potential. During the incipient plasmolysis, when a cell is kept in hypertonic solution, this is plasma membrane. When a cell is kept in hypertonic, that means concentration in the surroundings is maximum than more than cell. First, the plasmolysis, shrinkage of protoplasm occurs only in corners, then the shrinkage extends and the total protoplasm get shrinkage. So, the initial stages of plasmolysis only in the corners is called as incipient plasmolysis. So, here the question, what is meant by incipient plasmolysis and how can you prove incipient plasmolysis? The question is, during incipient plasmolysis, the protoplast is intimately associated with cell wall is wrong. The protoplasm is completely withdrawn with the cell wall is wrong. The protoplasm is withdrawn from the cell wall only at the corners is correct. The protoplasm is withdrawn only from lateral walls is wrong. So, correct is the protoplasm is withdrawn from the cell wall only at the corners. Third option. Capacity of water to do the work is reduced by. Capacity of water to do the work depends upon the free energy present in them. When you add some solute and increases its concentration, its capacity decreases. When you add some metric component, what is metric component? Whenever it is added to water, it absorbs water at the surface level because of hydrophilic colloids. It does not dissolve in water, but decreases the free energy. So, whenever free energy is decreased by adding hydrophilic substances, the activation or what you call capacity of water also decreases. Why? Because the capacity of water depends upon the free energy of water. So, not osmotic pressure, solute pressure and hydrostatic substance. So, solute and hydrophilic substance are the two components which makes the water to decrease their capacity. So, capacity of water to do work is reduced by solute and hydrophilic substance. Hydrophilic substance means it does not dissolve in water, but it absorbs the water at the surface level and reduces the free energy. Answer is 4, 2 and 3, that is solute and hydrophilic substances. This is always has negative value. Chemical potential, osmotic potential, metric potential. Osmotic potential means solute potential. Solute potential means the concentration is more, free energy is less. So, free energy is less means negative. Metric potential. Metric potential means there are some metric components. Like wooden pieces, seeds, that too dry seeds, cellophane or some filter paper, something like that. When these are added to water, they don't dissolve in water, but they absorb the water at the surface level and reduces the free energy. If free energy is reduced, the value will be of negative value. Why? Because pure water is 0, less than that is always negative value. So, which has definitely negative value means not chemical potential, but osmotic potential and metric potential. So, answer is 3. 2 and 3 definitely have negative values. This is responsible for metric potential of a cell. This is responsible for metric potential of a cell. Protein, sodium chloride, copper sulfate. Sodium chloride and copper sulfate are solutes which are added to the water, which dissolve in water, increase concentration and reduces the what you call water potential. Protein absorbs the water at the surface level. Definitely proteinaceous seeds show more inhibition than starchy grains. So, protein shows more inhibition. Inhibition is nothing but metric potential. Absorbing water at the surface level, not dissolving water is metric level and metric potential. So, protein, sodium chloride, copper sulfate. Sodium chloride, copper sulfate comes under solutes which increases solute potential. So, this is responsible for metric potential of a cell means protein. Answer is only 1. Only protein controls the metric potential of a cell.